Well, it's a great honor for me to introduce Dr. Alexandre Antonelli, who many of you know already. He's Director of Science at the Royal Botanic Gardens, Kew, in the United Kingdom, where he leads the work of about 500 scientists working in more than 100 countries, and is responsible for the world's largest collections of plants and fungi. He is also professor in biodiversity and systematics at the University of Gothenburg in Sweden, visiting professor at the University of Oxford, and distinguished scientist at the Chinese Academy of Sciences. Alex is a Brazilian botanist studying diversity, distribution, evolution, and conservation of species, and developing methods to speed up scientific discovery. He has published over 230 peer-reviewed scientific articles and book chapters, and has been continuously named on the Web of Science's highly cited researcher list since 2020. So welcome and thank you, Alex, for this talk.
Thank you.
Oh, yeah.
All right, great to be here. Thank you so much, Adam, and thanks so much everyone here from the university, from the Lancaster Botanic Garden, for having me here. It's absolutely a huge privilege and amazing honor to be here with you today. And I know this is a congress about monocots, so I will talk about monocots. But I just also want to say, I know that some people were trying to take notes, so it's fine to take photos, just as long as you Photoshop them afterwards, and you can pick, you can just put some hair or make maybe younger. You choose it, but it's fine. And I do want to start looking at how monocots fit into the greater picture of life on this planet and the amazing diversity that, as far as we know, is the only planet that is inhabited. And what does it mean in terms of understanding and protecting our planet and its life? So what I would like to start with is to talk a little bit about our great embarrassment. And I'm very embarrassed about the fact that we know so much more about the surface of the moon or Mars than we know about life on Earth. But then I also want to say, who cares? Is it something that only we, in a monocot conference, are troubled by? Or is it something that perhaps others also should be thinking about? And then I'll talk about this concept of global dark spots. And I'm not sure anyone here has heard of it before, but I think that's a nice and useful framework in terms of understanding our diversity and where it's found. And finally, I'll go from the global to the local. So understanding how we can use that overall global framework to actually do on the ground biodiversity science and conservation. So what is the great embarrassment? I don't know whether you are embarrassed. I'm a bit embarrassed by the fact that, after 250 years, even more than that Linnaeus started his journey to describe all species on this planet. We know somewhere around 10% of all species. And it really troubles me when I speak to my mother and she really doesn't understand it. We spent hundreds of years really trying to document the diversity of, for instance, just plants, and we're still far away from having that number correct. And when it comes to some other groups like fungi, it's just a nightmare. We perhaps know less than 7% of all species. So I think it's a really striking pattern and something that, even though we think we know what we're talking about, in most cases, it's just like playing a concert with only 7% of your keys in the keyboard. I don't think that even Mozart would do a good job in doing that. So I think we need to be a bit humble about the fact that we know so little, and there's so much that we know we don't know, and how that affects our decisions in every day in terms of conservation, in terms of mapping, for instance, evolutionary patterns and processes. So you see the plans here. We have an estimate. We published last year the first verified world checklist of vascular plants, where every single species has been assigned a geographic region where it occurs. And the estimate that we have here of 400,000 species, and perhaps about 50,000 more, but there's a big question mark there. And you see that the uncertainties are even larger for, as I said, the more diverse groups like fungi and invertebrates. So you see the numbers in black here of the number of described species, and what we think, but it's really just a guess of the number of species we have in total. And we are really curious about plants because they're so important to almost every single ecosystem on this planet. And without knowing how many there are, that really puts us in trouble sometimes. We compiled last year an assessment of how that knowledge has grown over time. And you can see here in this plot that for each of the different regions across the world, the number of described species has steadily increased up to now. So when Linnaeus started, that's in 1753 with the first publication of his plant account. Since then, of course, we've considerably increased our knowledge. But as you see, there's no place this curve is actually flattening out. We can't see any tendency that this is actually, where we're getting our job done. So I think that there's no question here that no one will be, bored by being a botanist until they retire and long after that, because there's so much we know that we don't know. But there's a problem here, and the problem is that we're describing new species to science, but a large part of those species are threatened to go extinct. So at Kew, with our partners, we've assessed that about 45, so almost half of all plant species, especially flowering plants, are likely to be threatened with extinction. But it's just, if you see here in this plot, what happens is that the species that were described at this particular year, for instance, what is the conservation threat, the conservation status of them today? So you can see here that the species that were described in about 1900s, less than about 25% of those are considered to be threatened. But when it comes towards the present, which is here, that proportion is very different. So today, about 3 in 4 plant species that are newly described to science are threatened. From day one, about 77% of all species are going to be threatened. And it's not that strange because we know that the conservation status is very dependent on the geographic distribution of species. So many of those species that you guys have described, I'm sure, are only found in very small geographic areas. So if someone comes and builds a new factory, or there's a volcanic eruption, or something else, of course, those species may disappear forever. And as I said, that number is very striking. So 3 in 4 of those newly described species, which is really something that I think we have to think about, because that curve, as we just saw, is going up. So if we wait 10, 20, 30 years, that number will likely be even larger than that, which means we have to speed up our rate of species description. And the problem is, if we continue with business as usual, that won't happen. For fungi, for instance, we estimate that it will take between 750 years to 1,000 years to describe all those species that we haven't yet found. And that's something that, of course, we don't have because those species won't be there anymore. So we don't want to be studying our herbarium specimens, for instance, knowing that these species may no longer be in nature. So who cares? I think we care, right? I mean, I can assume that everyone sitting here, we are, if you're like myself, just passionate about nature and think that that's the best thing to do in life, to be, looking at a specimen or being out in nature and collecting plants. And of course we do, but there are more people who do that too. And the fact that we've seen since I was born the destruction of about 25% of all tropical rainforests is not something that only biologists are concerned about, of course. My family in Brazil, they are really struggling to get clean water. They are really concerned about the effects of landslides, for instance, when there's a heavy rainfall. And that effect, of course, is much more severe in poor countries around the world. So habitat degradation or deforestation in this case is the #1 cause of biodiversity loss. That's the main driver, but there are others as well. And just last month, you may have seen on the news that Texas was burning. So that's an image from there. And we are just in early March now. And last month, we have seen that it was the warmest February ever since we started to measure. And you see here all the Februaries are plotted on the scale. And those records are being broken every single month now. So we are living outside anything that has previously been known. And of course, climate change will have an impact on every human being's lives, but also on the species we care about. So the combined effects of climate change and habitat loss are, of course, something that should be a matter of concern to all of us here, but everywhere. And it's, again, it's not something that only we care about. At the World Economic Forum earlier this year, you may be aware, but they put this report showing the most important risks to our global economies. And the longer-term risks is actually interesting because it's become more and more green towards the top, which means those are environmental risks. Compared to anything else, like disinformation, migration, pollution, cybersecurity, those things that we're talking a lot about now, but in the longer term, we do know that environmental destruction, biodiversity loss are really at the top. And biodiversity occupies actually number 3 and 4 here. We think that at this point, I would argue that perhaps it's even more important because species that go extinct, they cannot come back. And it's also interesting to see the difference that biodiversity loss impacts on people's lives. If I'm hungry, if you're hungry, just go opposite. There are so many restaurants around us. We are very privileged to be able to afford eating and buying medicines in a shop. But of course, not for thousands or millions, but for billions of people around the world, that luxury doesn't exist. And we know that in large parts of sub-Saharan Africa, Southeast Asia, parts of Latin America, the importance of nature and plants to help people deal with ailments is something that is absolutely crucial for their survival and well-being. So recently, we put together a comprehensive list of all the known species of plants used by humans. So there are about 35,000 different species that we know are used as medicines, as source of food. And that's something that we also have to think about, the importance of plants and plant diversity for the lives of so many people around the world. And that's a work led by our colleague, Sam Pironon. So it's a really dire picture. I'm sure no one here is not aware of those challenges, but I'm also very optimistic, actually. I did have the chance to participate in the COP15 meeting in Montreal, and I was very surprised by the fact that so many nations, almost 200 countries, came together to discuss and agree on a very ambitious set of agreements and targets up to 2030 and 2050. Actually, I think it was interesting because one of the reasons why I think they agreed on that is that you could see very few presidents. This was mostly the environmental ministers, so I don't think they really realized the costs of those agreements. But that was a good thing because it's really a chance of turning the tide and bending the curve on biodiversity loss. And you may have seen the terms of that agreement. I personally think that was the most important meeting in human history, because we know that if we don't get this right now, without exaggeration, we may really lose so many species, so many habitats around the world, and we have one last chance, I think, to turn things around. But some of those things do require our knowledge and the expertise that you guys have. So for instance, I'm sure all of you have heard about the 30 by 30 commitment, which is really focusing on a quantitative measure about what we want to protect and where and how much. And there are really good, it's a really good and easily communicated aspect that is included in the agreement. But there's some caveats. And for instance, by 2030, which is 6 years from now, hurry up, we have to bring the loss of areas of high biodiversity importance closer to zero. And also at least 30% of all terrestrial areas of particular importance biodiversity are conserved. Can anyone here tell me what areas of biodiversity importance are? I actually don't know. Is that a lot of species or lots of rare species or endemic species, threatened species, economically important species? What species? Plants, animals, birds, amphibians, insects? There are lots of interesting aspects which, of course, were not fleshed out. And the devil is in the detail. So that's, I think, where we as a community can really contribute to informing and helping our policymakers and politicians to make the right decisions so that the areas that are protected are really comprising not only charismatic birds, perhaps, but also plants, also monocots. And it's really up to us to speak out rather than waiting for them to knock on our doors. Right. But as I said, we on one hand have those amazing discoveries, and we're describing new species all the time. And in this conference, we've heard so many great discoveries and findings from scientific knowledge to the applications of the knowledge. But on the other hand, we have this huge gap and challenge ahead of us of identifying, describing, and studying species around the world. And how do we do that? And that's where we've been introducing this concept of global dark spots. Because the collections that you make when you go out in the field, and I make, and my colleagues make, are really the primary source of information about life on Earth, right? It's really where we can attach a specimen species to a place and a date. And that really helps us understand how things change over time, how they may change in the future due to climate change and so forth. But of course, you know that there's a lot of biases here. So even though there are hundreds of millions of records of georeference plants, for instance, we also know that it's a hugely biased data set. Because we go to places that are easy, we pick up flowers that are at high height or have fallen on the ground, because we're just humans, right? And because it's not very easy to go into deep forests for days and days without all the food that you need. So they're all good reasons, there are lots of good reasons why the information and knowledge we have about plants and other organisms is very biased. But it's a problem as well, because it really doesn't allow us to get a deeper and better understanding about all that diversity. So already several years ago, for instance, like these and many other papers have shown that those biases are everywhere. So this just shows in different colors here. So blue means that there's a lot of species, but very few collections, right? So you see here that a large part of the world is blue, which also means that people like myself and my colleagues in Northern Europe and parts of North America should perhaps not always be working in the countries there, but really working where the diversity is greatest and the needs are the largest. So what we did was to bring together about 200 scientists last year. And there are several people here involved that I know in our report that we published from time to time, about 2 to 3 years, called The State of the World's Plants and Fungi. And we had actually a single overarching question about where do we focus our efforts moving forward? Because looking back, that map, which really shows how much biases we have, we need to do a better job in terms of coordinating our efforts, in terms of filling those gaps that matter the most. And this was a big effort, very international, and really involved scientists from many different countries and institutions, as you can see here. And one thing we did was to use our historic knowledge about our accumulation of knowledge over time to predict what's happening in the future and where to focus. I'll give you an example. I'll just show you a bit how the method works for identifying those dark spots that I mentioned. And as you can see here, there's a random species, in this case, a Caromia. I'm sorry, it's not a monocot, but it's from Madagascar, so that's only found in Madagascar. And what we can see here is that since Linnaeus started his job, of course, many people before him had also been working on naming plants, but anyway, let's take Linnaeus here. And the number of species has accumulated over time, right? So this species was described here in 1890. So there was a gap, which is the gap or the time for description. So how long did it take before we kind of started the job within the modern nomenclature to describe the species? But then from that description until we actually had a knowledge about where that species was found, which is, as you see here, Madagascar. So it took a bit of a while. So that's the first geolocation, the first record that we have in our databases with some sort of coordinate, which we can then use in the models. So that's the time of geolocation. And we also know that diversity and the exploration of diversity is not random, right? As I said, we are, we're going to different places because of accessibility, but that's the same for when it comes to the diversity of species. We kind of know, if you're a botanist, you look at a mountain that, oh, I'm sure that there'll be lots of species there, and then you look at another place, I don't think that'll be very interesting. So because we know, for instance, that things like environmental factors, so temperature, humidity, precipitation, we know that, if a species has a big range, will probably be easier to be found rather than a species that is very, narrowly distributed. If people use plants for, medicine or other purposes, that's probably going to have an impact as well in terms of description, and also life forms. As I said, we walk in a forest like this, and then we just see things at our height. We don't necessarily look at things that are 30, 40 meters above ground, or perhaps even on the ground. So all those things may influence the likelihood of a species being scientifically described. So that's based on that model, we can actually look backwards and look forwards in terms of where are we most likely to find new species and new records. So the first result here is the taxonomic knowledge gaps. And this is often described as the Linnaean shortfall. So the Linnaean shortfall is about describing all species to science. And you see here in the different colors that this is a map of where we believe, based on those trends in the model, there will be most species to be found in the future. And we can do the same thing for the geographic gaps. So basically, if a species is known from Costa Rica, for instance, how long will it take before it's known from Panama as well, or from Colombia? So those are species that are already known, but their geographic range hasn't really been fully documented. So those are the places where you're most likely to find species from neighboring or other regions as well. And I'm not sure if you're familiar here with the divisions of the countries. It's called botanical country. It's at level 3 in an internationally agreed spatial categorization. So some countries here, like the whole country is one of those botanical countries, and sometimes like Brazil, there are several different botanical countries within Brazil, just the way that the data set is structured because of floras and different reasons. But we can combine those two, and we can produce something that we then call a dark spot score, which is basically the combination of those two factors and what's the total value that those factors will give to a particular country. And as you see here, the number of, all the countries can be ranked. And if we put a certain threshold just to match the biodiversity hotspots concept, so we don't have like the whole world, we need to put some sort of limit here in terms of how many dark spots. If you use the same area as biodiversity hotspots, we'll get this set of 32 dark spots. It's not set in stone, and there are different ways of doing this. I'm not going into the details here, but there are different ways of prioritizing. For instance, if you want to prioritize countries that have the most economic challenges, and perhaps you want to favor them and so forth. But overall, you see here on this map, the dark spots, which are the areas where the global community, such as this, could be considering in terms of moving forward and increasing our knowledge generation in those countries. And I want to just, again, talk a little bit about what does it translate in terms of action, in terms of the global, from global to local. And I just want to mention 3 examples here. One of them, of course, from my Tico friends, Costa Rica, and then Madagascar, and then a little bit about Southeast Brazil. So let's go from global to local. And I think it's really important because some of those analyses are just like very, large scale, and people don't really understand the local and regional considerations and the importance of really understanding the complexities that are involved in doing field work and really working with our partners in very equitable and iterative ways. So the first one is this beautiful country, Costa Rica. So I had the chance to go with Oscar and Diego to look around a little bit. It's my second time here in Costa Rica. So this is just a few days ago in Cerro de la Muerte. And it's a beautiful area with a lot of forests, wonderful species all around. And I think that by just, understanding the diversity in those mountains and other ecosystems in Costa Rica, there have been huge advances in terms of our understanding about evolution, about biogeography, about phylogenetic relationships. So because it's a monocot conference, I just picked up a couple of works that we've done on monocots with my colleagues. And this is a work led by our colleague, Angela Cano from Colombia. And she's been working a lot on palms. And, back to Maria Jose's comment, there hasn't been a lot of palms in this conference, but I do think that palms are incredibly fascinating and important. And so what we did here was really to look at the evolution of 3 major clades of palms with a densely sampled phylogeny, reconstructing their biogeographic history, and then finding, as you see here, the different colors. So Costa Rica and Panama here, Mesoamerica is the D, that's the orange. Is that orange? I'm also a bit colorblind. But you see there's a lot of orange here, and that orange is mostly concentrated towards the terminals, right, of the tree, like the leaves. And what we could find here is that there was a very recent and very rapid speciation of palms in this part of the world. And we know that. There are lots of fossils here, for instance, in Costa Rica, which show that this place was largely covered by water about 10, 12 million years ago. So it's a rapid evolution of the landscape, which also provided many new niches for evolution to take place. And this is due and thanks to the incredible work that people have done here in Costa Rica in these regions to document and sample the diversity of palms. Another work that you'll be familiar with from Oscar's talk this morning is on orchids, and this paper that was just published recently in New Phytologist, led by Oscar. And I just want to point out that based on this, again, huge phylogeny and very densely sampled as we can today, of course, the many species that have not yet been included because it's such a hugely diverse group. But what I want to point out here again is this amazing diversity and the fact that where we are right now is actually the stage of a rapid radiation for orchids. And globally, at least according to those analyses we did, this is actually the place with the highest speciation rates across all the family. So of course, this could be biased. A lot of people here have been sampling, so there could be all kinds of refinements of those results. But it's really exciting that we may be in the place with the hottest diversity for orchids in terms of speciation. And again, this is really based on the incredible work that people do and spend days in the forest and collect those species, and that contributes to our understanding of evolution and diversity. And I also want to make a huge thank you and congratulations to Lancaster. I talked a little bit about this in my speech Monday evening, but I'm so impressed because I was here almost 20 years ago, and there was not much there to see, but the work that the Lancaster Botanic Garden has been doing, especially I think in orchids, it's incredible. So we were looking at the labs, and I hope that many of you will take the opportunity to go there on Saturday as well to see the garden. But it's a major center of orchid, orchid research, and I think within Latin America it could be like a major hotspot of orchid research and education and training collaboration. So hopefully many of you here in this room will want to link up to the researchers there, including the great work that Lancaster did. The paper is publishing, and I think internationally it's one of the best journals for orchids, and it's a really great thing that you guys are doing to really communicate in a very open way the results from many different works in orchids. My second place, I said I would go through three different regions, is this place. Has anyone here been to Madagascar? Yes? Not many people. Maybe you've seen Madagascar, the penguins of Madagascar? Yeah? If you've seen it, just forget everything. It's not quite like that. It's a great place. I've been to many places in Latin America, Africa, but it's like, on one hand, it's wonderful. You see the lemurs jumping around, you see those fantastic trees. It's really of incredible diversity, very highly endemic. On the other hand, it's terrible because it's the poorest country not at war. You have to travel for 12 hours before you see like a small fragment of forest. So it's this schizophrenic life to some extent, and it's really interesting system. So we at Kew have been working there since 1986. So we have 65 members of staff working there, and that's why I've become so interested in Madagascar as well. So what we did recently was to bring together the knowledge about how are we accumulating knowledge on the diversity of different groups over time. And again, just like the global picture shows, there is nowhere any of those curves, and those are number of species over time for ants. Sorry, it's a monocot conference, but ants, reptiles, amphibians, and then plants, including monocots. And those curves are all going up. So the more you do taxonomic work, the more you find. It really pays off. It pays off in terms of the all the people from Madagascar who have got MScs and PhDs and so forth, but also in terms of our knowledge. And it's not only something that is interesting for biodiversity scientists like ourselves, but also for conservation. So what we also did was to assess the threat to all those species. And when it comes to plants, for instance, you see here that anything that is below this black curve here is bad, right? So all those species here, you see those different groups, all those species are threatened, and all those ones that are above the line are okay, right? That's what we think. But we also, for instance, let's focus on the monocots. We also did something that we think is going to be very powerful in the future, which is to use AI, so a methodology that uses assessments to predict the conservation threat of species that have not yet been assessed by experts. And what we can see here is that for monocots, we had 822 species, is that correct? That were assessed by experts. And we were able to add over 1,100 species that no one has ever assessed, but based on our AI methodology, we could also look at them and actually see that they were slightly more threatened. So we can have not a replacement for expert assessment, but a tool to prioritize which ones we should look at next. And of course, we would be very keen to look at those species here, which have not been assessed, but are very likely to be threatened. So we need to understand where are those guys? What can we do to protect them? And I know it's a monocot conference, but I just want to point out the ferns as well, because people thought ferns were fine, but they're not, because it's a bit difficult to see the colors here because of the projection. But you see here, that's the AI assessment. So actually, ferns are just as bad as any other group here. So basically, people have just assessed a few species they could find and thought that most of them were okay, but actually, it's not that. And I think that there's some really good opportunities for using AI and some other machine learning methodologies to help us crack on with the work of describing and also assessing the threat of species. The other thing, and this is for my monocot friends, I'll talk about two monocot groups. The first one being grasses in Madagascar, over 500 species, about half of them endemic. This is work mostly led by my colleague Bat Sova from Kew. And based on the species we know, we can do species distribution models, and we can predict the diversity richness, so the species richness across the island. So you see here, that's the curve and the number of predicted species. And then you can do a comparison, like each cell, how many samples we have, and how many species we think there should be. And you can see here that what you want to have is a lot of dark, very dark here, which is a good match between how many you've sampled and how many species there are, okay? So that's okay. It's just like two small cells here that are a bit off. We can go there and do more research. But then we have palms. And, again, palms are really important for all kinds of reasons. And this is the predicted diversity of palms across the island. And this is, the same analysis, but showing here lots of, like blue, for instance, which means that there's a lot of species, so that's the species richness. There should be lots of species here, but no one has bothered to collect them or has been there to check if they're still there. So this is really areas where we should go very quickly to. And those are areas where there are lots of collections, but actually not so many species. It's fine. It's a little bit of waste of time, but, perhaps we did something else there. And this is a way of, zooming in into a dark spot, because the dark spot analysis we did at a global level can only get us to this, botanical country scale. But of course, we need to understand at a much finer granularity, where should we do field work, where should we collect specimens, and how do we do that, and which taxa we do that for. So we can do that by doing this methodology. So my third and last place is where I come from, and that's the Atlantic forests of eastern Brazil. I just need a bit of water. So basically, a couple of years ago, my wife and I, we just decided to put everything we had almost to buying some rainforest. We had 200 pounds in our account afterwards. I don't know whether it was very wise, but we are passionate about doing something on the ground. So we created this charity to really promote research conservation in the Atlantic forest, and established this project here, which we call Araçá. And that's just starting, but that's the Atlantic Forest Research and Conservation Alliance. And what we're trying to do is really great research, conservation, and training. And I asked about Madagascar. So how many of you have been to eastern Brazil, the Atlantic forest? Yes, hands up. Good. Almost half of you. So it's kind of similar to some extent to the cloud forest we have in Costa Rica. But there's a difference, which is that most of it is gone. So it's a global biodiversity hotspot. It's a dark spot, as we mentioned before, but it's also mostly gone. So there's some different estimates. Around 8% is perhaps what we have in terms of good forest. And you see here, everything that is in red is basically gone, and some dark green spots, but not many of those. And still some deforestation taking place, but it's a bit better than in the southern fringes of the Amazon. So we are really keen to do now a major conservation and restoration effort to create a biological corridor of about 200,000 hectares, and doing that in partnership with the Atlantic Forest Trail. So this is the reserve where we have our headquarters of the work, and this in white here. Oh, sorry, in white is the Atlantic Forest Trail. So everyone heard about the Appalachian Trail in the US, right? Brazil has never had a trail, like a long trail. So that's the first effort of creating a 4,000 kilometers trail. And the idea here is to reforest about 50 kilometers on both sides of it, to bring ecotourists, but also to start restoring. You have to start somewhere. So what we're doing here is to use AI, again, in this case, a methodology we developed recently to identify the most important areas for conservation and for restoration, so we can optimize the combination of areas to be protected, so that we can get as many species protected over time as possible. So that's how this methodology helps us on a landscape to identify those areas. I'm happy to talk about this later in the break if you want. And we are also very keen to use new technologies to help us doing that, because we're talking about the speed of discovery, and we can't just rely on us doing traditional inventories everywhere. So we're now flying over those 200,000 hectares with airborne LiDAR systems, and we're going to do hyperspectrum and multispectrum scans of the forest, and using different technologies that we are using, for instance, for recording animals with camera traps and with audio recorders. We're capturing spores and pollen, and we're sequencing that and matching to database. So new technologies that are really helping us to quantify at a much greater scale and faster scale than only traditional inventories, and matching those two. So this is just a picture of how it may look like. That's a 50-meter plot that we just recently established. We have a very high-resolution LiDAR scan, so that's a laser scan basically, and we can really identify at a very minute level the diversity that is found in those forests. And we'd like to now to do botanical surveys. So please, my friends from Brazil, help us to get a name on all those trees and epiphytes. And we are also doing a deep dive into one tree, which I'm absolutely obsessed about, and I dream about it almost every night. I can't stop thinking of it. It is a fig tree. It doesn't look that incredible from below, right? That it's big, it's large, it's fine, but it's not that extraordinary. But we've been keen to understand how many species do we have on that tree, right? How many monocots, how many dicots, how many fungi, how many of everything, basically, mosses, lichens. So we've set up some scaffolds around it. We spent months now collecting every single species on this tree, taking vouchers, and we are sequencing the DNA of lots of different places. And my assistant, she's been climbing all over those branches and doing really crazy collection, but very safely. We're paying quite a lot for the guys who manage the security here. But we're also bringing those machines there. You see that machine on the scaffold, which is a LiDAR scan. I just showed a LiDAR scan before to you. And that's what it can help us do. We can get a fully three-dimensional tree, and we can really fly over this, and we get a very highly accurate image of every single species on this tree. And this is at a millimeter level, and we're very impressed because, as I said, we're mapping every single species of epiphytes, and we're really keen to understand how this formed. But we were also surprised because we thought it was one tree until we actually saw there's a second canopy, and it turned out that it is a fig tree in the top, and it's a Sloania in the bottom. And we're really keen now to also build a tower around this, a permanent tower, and a canopy wall, so that researchers, students can research this tree for many, many years to come. And together with Oscar and colleagues, we're also trying to innovate on the use of new technologies, including like nanopore sequencing, to do rapid species discovery and identification. So for instance, recently we did a one-day sampling on Monday, and then we put the things for sequencing in the evening. And then after 3 days, despite 3 power cuts, we got at least 1 new species of fungus and 2 new species of moths. So I think that this is a really good opportunity to also speed up discovery of species. So this is the place, the house there. We're trying to turn this into a Barra Colorado for Brazil. and I hope you understand what I mean, to have a base for long-term studies, monitoring great research. So please do consider this as your next stop in your fieldwork and of your students as well. So we are now, we just demolished the house. We are building a facility that will accommodate up to 24 students and researchers. And also we'll build eventually some labs. We need to do some fundraising for the labs, but I think that'll be a really great setup for a lot of research and work there, including monocots. So just before I finish, and that's time for you to pull out your phone to take a couple of pictures. And the first one is to make a call for you to become a co-signatory, if you want, on the 2030 Declaration on Scientific Plant and Fungal Collecting. So basically, I told you that we had this big symposium. We had over 200 scientists from 30 different countries, 100 institutions, talking about where do we go next? Where do we prioritize our collecting efforts so we can fill up the most important gaps? And we've since then worked with everyone and developed a set of 5 commitments that we'd like to do, both individually, so each one of you, if you want, and please, urge you to consider that. You can sign it if you agree with those, and then your institutions may also sign that separately. So the first one is about using evidence, evidence-based collection strategy. A lot of us, we go to a place just because we have friends there or there's a tradition. There are all kinds of reasons why people have been collecting where they have. But we think that we need to be a bit more strategic in terms of really going to the places that are most important in terms of conservation, in terms of the expected diversity, and the dark spots framework is a suggestion for that. The second one is about strengthening local capacity. It's about really making sure that any work is not doing it as a helicopter scientist. It's about engaging with the local partners and really understanding, listening, and responding to demands. And it's about leaving collections, so the plants and fungi you collect in the country, at least, and also to build capacity, both intellectually but also physically, if possible, in those countries and regions which are really the important ones for collecting forward. The third one is about collaborating across taxa and disciplines. And I'm afraid to say that it does mean that monocot people have to perhaps help others, non-monocot people, to collect things that you find. And I think that will be very important in terms of cross-disciplinarity and better understanding the biodiversity of ecosystems. And that may also include, for instance, fungi, or perhaps bringing up along a zoologist friend, so that we really understand a bit better the interactions that exist among plants and other organisms. The fourth one is about collecting for the future. So both making sure that whatever we collect is going to be as useful as possible in the future, for instance, collecting silica gel samples or other accelerated data, but also making sure that the collections are safely stored and useful for the future, for all future, which of course is absolutely critical for any further research. And finally, it's about sharing the benefits. It's about sharing the knowledge that is derived from this work, especially with the communities and scientists, students in the local communities where the field work is done. So that you can download the declaration. Of course, it's several pages fleshing out those principles and the rationale. But if you agree to that, please consider putting your name on this and sharing it with your networks. And my second one here is a call for papers. We are just in the process of finishing our digitization, progressing our digitization of 8.5 million specimens that we have in the herbarium fungarium. And many other organizations have also done that or are in the process of digitizing their collections. We are keen to understand what can we do with all that data. And I'm sure you have ideas here about, how has digitization, so the images and the information of geolocation of specimens can help advance science, conservation, how can the use of new methodologies, for instance, help in that. So please consider submitting an abstract to this call. And this will also, so it will be a publication in New Phytologist and Plants, People, Planet, a joint collection, and also fit into the next State of the World's Plants and Fungi in 2026. And every time we publish that report, it reaches about 500 million people. It gets a huge amount of attention, it influences policy. So it's not only a scientific exercise, but it's also something that hopefully will help other countries get funds for digitization, for instance, and for advancing science. And that's it. I just want to conclude. And the first thing is, I don't think I have to convince you, but I hope I have convinced you otherwise, that biodiversity science, including taxonomy and many different aspects we are covering this conference this week, are more important than ever. It's really urgent we do that. That speeding up the discovery and documentation of species really requires expertise, so people who really know their stuff, but also innovation. And that's where I think AI and remote sensing, the integration of knowledge and technologies is, I think, the most powerful route forward. And finally, I think we all agree that the task ahead is huge. You have lots of ideas in your heads about things that need to be done, lots of species we don't yet know where they are, if their names. So please let us try and join forces, because I think I'm very optimistic. I think the way ahead is great, and it's so much more fun to do that together. Thank you.
Thank you very much, Alex, for the wonderful talk. So, if anybody has questions for Alex?
One question about digitalization of collections. I've been working with GBIF and all kinds of databases, and unfortunately, probably half of the specimens are misidentified. Sometimes it's simply the label is not correctly read, but in many cases you have a combination of all kinds of similar species which under the wrong name. And I have not found a way to be able to get the owner of the species, of the specimen, the herbarium or whatever it is, to correct it. Do you have any ideas of how we could get up a system so that those who are working with the material can actually, without having to travel and put a label on the specimen, get it corrected?
That's a great point. Thanks, Eric, and really important one. And I think that's one of the many great things about digitization, because, for instance, if you go in the field and collect 5 specimens of the same tree, and then you send it to New York, Missouri, and Costa Rica, and Kew, we need to think more about digital labeling rather than only physical labeling. So we do discuss this a bit in that declaration as well. We think that for now, it's usually beneficial to have both. So we need systems to enable that to happen. And we know that, as you say, maybe 50%, perhaps 40% of all specimens may be misidentified, misclassified. So I think that the digital assets are a way of doing that. I know GBIF is actually it's not a database, it's a portal. It really reflects the data sources that are providing information to GBIF. I'm sure there will be people here who are much more experts than I am, but my understanding is that they've been challenged, because I've approached them in exactly the same questions. When we clean a lot of data, we want that clean, that information to feed back into the sources. And I think I agree with you. It's not really happening at the moment. But there are some efforts. I know in the US, you have IDIG-Bio in Europe, we have DISCO. So there's some different international efforts to bring those facilities in and improve those over time. So it should be doable. I'm not the one programming that, but of course that has to happen. So it's a really good point. Thank you. Sorry, I didn't really answer that, but yeah, anyway. Sean.
I really shouldn't be making this point, because Tom Croat made it very eloquently at the end of his talk about the world is basically burning or being churned up. And the way that permitting systems work with governments who want to understandably protect is actually the antithesis of what we should have. And I know you probably can't comment on this as the director of Q, but maybe you could give us your personal thoughts on how we could change that a bit to make it more facile for people to make collections around the world.
So if I really get your question, I think on one hand the word is burning. On the other hand, we want to generate more data. We need to learn more. Is that more or less what the dichotomy you're describing?
And things that are put in place for a good reason, permits, CITES, and so on, are actually having the opposite effect, because they restrict discovery, science, and so on. And maybe Tom could comment on that if he wishes. But
right. So okay. So if the question is about the legal framework, I think it needs to be workable, and it should really concentrate on the problems rather than punish the people who are actually trying to do the right thing. So in a sense, you should not be guilty until someone proves someone proves you the opposite. But I think it's a bit the opposite, isn't it? I think there's a bit of suspicion, and understandably so. And we're working in countries like Ethiopia, for instance, where it used to be easier to do international collaborations, but they had some really problematic histories with, Dutch company trying to make patent on teff, which is a very traditional crop there. So I think there's sometimes reasons for that, but you're right that not everyone in the political spheres understand that actually most people, the vast majority of people trying to collect specimens are actually doing that in good faith for the common good.
If I may, Tom, again, just
What he's saying is that the difficulties of getting permits and to make collections is almost so bad that people are just not even trying. It's impossible, so they just give up. And that's counterproductive because it's easier for a farmer in Colombia to get a permit to clear a great big piece of land than it is for me to take a sample off of the fallen trees. Why should that be the truth?
I know, for my master's, I was going to collect in the Amazon, and it took me 6 months to get permits for collecting some branches, and you can cut down the forest. So at the end of the day, Lula would have to sign my papers, they said. And it was his first presidency. But I think, put that aside, I do think that we also have to understand how we can best engage with the local partners. And I think, in a sense, even me, representing an institution which is 265 years now, we can't just think about going, collecting, and bringing things back. I think that, yes, if there are duplicates which will make those collections safer and more useful to the global scientific community, yes. But I think primarily we have to be thinking about the building of local capacity. And we've been having those conversations here, the fact that in Latin America, for instance, in Brazil, in Mexico, Colombia, many places around the world, South Africa, there's so much more taxonomic capacity than there was just 5, 10, 20 years ago. So I think, in a sense, we have to also turn it around and look at how can researchers in wealthy countries such as mine really make the best possible contribution on the ground with the local partners, and perhaps they should have the permits, not me, you see. And I think there's some different innovative ways, but I do understand it's frustrating, but I think we need to work constructively, and I think sometimes there could be some solutions, but I'm very happy to continue that discussion.
If we stretch the time a little bit more, we have time for one more quick question.
I'll try to give a quick answer as well.
Early on in your talk, you showed the graph of rising numbers of species discoveries, but also corresponding rising numbers of threatened species. Given that most of the species we're now discovering are quite narrow endemics and probably don't have a vast impact on the global ecosystem, how worried should we be about the correspondingly rising threat of extinction of those species?
It's a very hard question. I don't personally believe in, like thresholds or boundaries, like safe planetary boundaries for biodiversity. I think that our knowledge on the medicinal properties of species, for instance, has shown that no one cared about that green mold on bread and everyone threw it away until Fleming discovered penicillin, which then saved hundreds of millions of lives. So I think there's also like the utilitarian, like the use of plants and fungi and other organisms for humanity, which could turn out to be absolutely critical in 20, 30, 50 years from now, like the next pandemic. Maybe the cure of that is hiding somewhere in the forest of Congo or, here in the Cerro de la Muerte. So I think there's like a human-centric perspective, but there's also like the moral aspect. I think it's a major societal failure to not protect our species. And I think we just been here, in this tiny last seconds of this planet's history, and I think that we have to do everything we possibly can to just hold our breath until we know better and can actually start valuing that biodiversity as a natural capital for people, but also for its own sake. My opinion. Thank you.
Well, thank you very much, Alex. Oh, do we have? Oh, I guess the conversation can continue.
I think Hanise had one, but we can do in the coffee break.
Well,
you're the boss. You decide.
I promise it's short. Thank you so much for your talk. This is very uncomfortable and, of course, very nice to hear all of these numbers at the same time. So important to hear all of these stats that we kind of ignore because we're, in our own world. But my question is, what are your thoughts about fungi? Because we have macrofungi that some people work on that, but we have so many more that are endophytic that we can't cultivate, that it's so many species that we do not know. What are your thoughts about how to improve the knowledge of that group?
I think fungi are the next frontier of biodiversity science. I think there's a huge potential. We are now just about to start sequencing the type specimens in our fungarium, which is the world's largest collection of fungi, about 1.25 million specimens, together with the Naturalist Museum. So there'll be 50,000 type specimens at Kew and others at the Naturalist Museum. We're not sure the money will be sufficient, but I think that will be one way of starting to speed up that work in linking environmental sampling, like the DNA, with the fungal diversity in our collections. But I think there's a huge potential there, and I think more people should consider mycology as a focus and target in their early education. But it's hard if all of us are, we're just keen on our plants, or, I think that we also have a task that we don't, we need to communicate that early on so that people get interested in them. I think it's super exciting, and the review paper, we talked a little bit about that challenge and how we can speed up things. And I think DNA is one of the many important areas of, speeding that up. Thank you.
Well, thank you very much, Alex, and
Thank
everybody
you.
else for the rich discussion.